Thank you for your introduction. Good afternoon, everyone, distinguished guests and audience. Before starting presentation, I'd like to express my gratitude to the chairperson, Dr. Kameda, and dedicated secretary, Ms. Natsuki Sato, and Professor Kosuke Kudo. And thank you for both making chance for me to meet with experts and dedication to ensuring the success of this event. Today, I'd like to introduce the interesting property of heavy water for the use of cell and organ protection in the field of organ transplantation. I have nothing to disclose. And our research interest is to save life of patients by organ transplant. But the critical shortage of brain-dead donors is a common issue worldwide. Expanding the donor pool is one solution. However, livers from donors with cardiac arrest or with fatty liver diseases cannot be transplanted safely. Therefore, we have attempted to safely utilize these unideal grafts. In this presentation, there is no data about 17O water. Instead, I'll introduce the biological activity of D2O in cell to animal levels. I believe it would be meaningful to simultaneously evaluate 17O water where the proton transfer occurred like normal light water, whereas such isotope effect is inhibited in deuterated conditions. In short, 17O water would serve as a tool to understand the dynamics of D2O, and conversely, D2O would serve as a tool to understand 17O water. Here we show the time course of graft injury in organ transplantation, so-called ischemia reperfusion injury. In the brain-dead donors, cold storage injury progresses slowly here. However, in cardiac death donors, the liver is compromised before cooling. Once the organs are placed inside the recipient bodies, they started to warm up. And since energy consumption doubles with every 10-degree increase in temperature, it is crucial to maintain a low temperature until blood flow resumes. However, when blood flow is actually resumed, rapid oxidative stress and inflammation are triggered and actually leading to massive cell death. In a healthy graft, the damage peaks within 6 hours, but the harmful responses may be prolonged in marginal grafts and causing further damage. Accordingly, suppression of the triggers of IRI before transplantation is necessary. The efficacy of organ cooling has been known since the 1960s, starting from whole donor body cooling and proceeding to the idea of organ cold storage. And the University of Wisconsin solution, introduced in 1988, is a groundbreaking solution, considered the highest in efficacy for over 36 years since its introduction. But it is still insufficient for the marginal donor grafts. So, what happens during cold storage of organs? We should know these things. It happens during cold storage. Firstly, the ATP depletion and reactive oxygen species production. And after that, cytoplasmic calcium overload and lactic acidosis, accumulation of lactate. It means glucose degradation is a carbon 6 compounds to carbon 3 compounds. It's thus for osmolarity became high. And after that, and these phenomena occurs, and simultaneously occurring these insults must be suppressed without burdening drug metabolism, especially in liver. And when I was graduate student, I was forced to read papers of 1 meter. The unit is 1 meter thickness in printed paper. And it was very sorry for the earth and jungle. But finally I encountered a faithful review paper according to it, papers. And according to it, D2O has these 5 effects. A hydrogen isotope, which was twice the mass of normal hydrogen, was discovered by US chemist Urey in 1931 and was named deuterium. After the discovery of heavy water, studies on its physiological actions started immediately. And generally, D2O causes cell death or growth retardation due to the stabilization of proteins by hydration of substitution of intramolecular H2D. In normal light water, hydrogen bonds between adjacent water molecules are constantly forming and dissociating. So hydrogen bonding is the cause of the unique property of water molecules. And here's the property of hydrogen bonding and normal water. In the mixture of D2O and H2O, HDO caused in mixture, HDO caused by equilibrium. For example, 30% D2O with 70% H2O causes finally about 49% of H2O and 9% of D2O and 42% of HDO approximately. So the total of 51% of water molecules contains deuterium. Accordingly, proton transfer and atom tunneling phenomena are suppressed to some extent in this mixture. So, whether D2O is protective or not was determined in mimicking cold storage and reperfusion. The hepatocyte cell line was subjected to cold hypoxia for 12 hours and subsequent normal culture reoxygenation at 37 degrees Celsius for 12 hours. And the lactate LDH leakage and MTT metabolism were assessed and revealed that the efficacy of D2O at concentration higher than 10% is effective to reduce cellular injury and maintain cellular viability. So, next we made a new preservation solution which named DSOL, which is modified UW solution, presented here. It contains 30% of D2O, and raffinose was omitted, and instead sucrose and mannitol was added. And sodium and potassium ratio was inverted. That's all. And hydroxyethyl starch was omitted. And Cardiomyocytes were subjected to cold storage in UW disol, and HSOL, which is disol, D2O dilated disol. Cytoplasmic calcium concentration was determined by fluorescent probe FLO4, and cytoplasmic calcium overload was provoked by the addition of 4-HNE in UW groups. That the calcium concentration was rapidly burst, whereas it was completely inhibited in disol stored groups. And further, disol HSOL group, which is disol D2O omitted disol composition, the inhibitory effect of disol in calcium overload inhibition was completely abolished. So we confirmed that disol D2O suppressed endoplasmic cytoplasmic calcium overload. In red hot, cold storage and subsequent transplantation was performed, and graft survival was improved in disol group, regardless of cold storage time of 24 hours or 36 hours. And after 7 days of observation, UW group showed a massive necrosis and fibrosis in pathology, and whereas disol group showed almost no injury. Here we show the graft appearance just after transplantation. The left column showed the transplantation preserved in UW solution, and the heartbeat is very slow. The heartbeat is very slow and with congestion and bad color. And the right column shows quick beat with good color, which is disol preserved groups. So deuterium oxide containing solution is effective in cold preservation of red hot. And next we assessed liver graft excision from rat. After cannulating the whole abdominal organs, after cannulation, the whole abdominal organs were flushed via abdominal aorta, and livers was removed and subjected to 48 hours of cold storage in UW solution and disol. Then the liver was perfused on extracorporeal perfusion, so-called an isolated perfused rat liver apparatus, which is extracorporeal perfusion with oxygenated buffer with glucose and some source of bile acid. And the results showed a liver injury at the end of perfusion was significantly improved in disol stored group, and apoptotic index showed reduction in disol group. So the disol is effective in liver cold preservation and reperfusion. And in that study, it's interesting to say that just after reperfusion, just after transplantation, the portal vein resistance was significantly lower in disol preserved group throughout the experiment observation period. And finally, the oxygen consumption rate and bioproduction which is the indices of liver function, is significantly improved. In other experiments, cold storage in disol and additional post-reperfusion treatment by hydrogen gas, which causes dramatic reduction in graft injury, shown by the LDH leakage or oxygen consumption rate or by production. When the liver graft was subjected to cold storage in UW and modified disol, graft weight change was significantly changed. This slide shows graft weight change from the end of cold storage to the end of perfusion. And in UW, treated group showed high value, which means swollen graft, and swelling of the graft was significantly inhibited in the good preservation solution treated groups. But it is interesting to say that the anti-edema effect is shown, but D2O and antioxidant deferoxamine is necessary, when both compounds is included. The anti-edema effect was shown. So we confirm the positive impact of D2O for the use of organ preservation, but we need some modality to trace the change of D2O concentration or D-atom movement. We'd like to evaluate water movement in the graft subjected to cold storage in D2O containing solution. And after transplantation, water molecules in the vascular space, interstitial space, and inside the cell could be distinguished by using stable isotope water D2O and 17O water. So we prepared to meet in the colleagues here with cutting-edge surgical technique. Just we prepared. So we'd like you to establish a method to simultaneously visualize and quantify both D2O and 17O labeled water. Please, Dr. Kameda, please. And thank you for your attention.